Other alkaloids I took were, I isolated their protobine and allocrotobine. This is one of the, I recommend this for anyone who needs very good crystals. We can tell you this is a very good and rich source of berberine in our country. And you are going to tell me that, well, you are talking about a rare or a vanished species that keep going to disappear. Well, I'm telling you that this is the same for papaver ruas, and papaver ruas is widely distributed in our country, and we can use it as a source for berberine. We did also biological evaluation. Our biological evaluation were directed either by the ethno-health practices around us in different areas, or sometimes we try to, let's say, try to do something else. For example, it's not shown here that we try to understand if this papaver ruas or papaver polytrichum has an analgesic effect. It didn't actually, we didn't have a sharp answer simply because it induces sleeping, and our rats actually went to sleep, and we couldn't differentiate exactly if they felt okay or pain were less or what. Concerning the acute toxicity results performed for female Strauss dowry rats, the highest tested dose was 2 grams per kilogram, and it's high. For us, it was good. We did not continue further simply because we have a problem with solubility. We stopped at 2 grams. Unfortunately, let's say we have some criticism, somebody criticizes, that's why don't you continue to higher levels in order to make sure how much the LD50. If you think in biology way, you don't need to have a very high doses. In order, if you take a cup of tea, actually, each time you have 1 cup, you may add 5 grams of your herb. And if you drink it, actually, as an average 70 kilogram person, you may use 15 grams a day if you use it for 1 day or 2 days or whatever. But you don't need it. We don't have to treat the LD50 as an absolute or like theta or epsilon or what. It's not a biological constant. It's fair enough for us to tell that this is non-toxic and this is perfect. Antiproliferative activity also were evaluated. Thanks to Dr. Rana Abdahab. Unfortunately, I was hoping to see her today. She helped us with that. The extracts, actually, all of them lacked any activity, while the berberine and papaverine showed antiproliferative activity against NCF7. Our results are good, but not super good like the literature is mentioning about these isolated compounds, 3 compounds. But they give us an indication that we have something to say. DBBH antioxidant activity, papaver polytrichum hydroethanol extracts was determined using DBBH assay. And the presence of antioxidant extracts leads to loss from the purple to yellow. Anyway, concentrations calculated as inhibition. If papaver polytrichum hydroethanol extract exhibited moderate DBBH scavenging activity, this will give us an idea if it is actually an antioxidant. People may drink it. One of the reports talking about one of province in Turkey taking these extracts for such reasons. Pancreatic lipase inhibition, IC50 were calculated for the exact conversion to orlistat. It was 0.123 microgram per ml. It is good and promising effect. And when we discovered that it has high concentrations of berberine, definitely we can tell, or we can correlate the results to say that berberine is responsible for such results. It may also indicate potential lipid-lowering agents. Papaver polytrichum is a natural source of berberine. I repeat, if papaver polytrichum is not available, we have papaver ruas still. There is papaver ruas available in our country. It shows a promising antibiotic natural source. It's also a potential antioxidant. We may think in the future also plan for further experiments in order to check for their ability to be anti-cancer for other types of cell lines, cancer cell line. Also, we can recommend that papaver species should be phytochemically screened for potential virus chemotypes. As I said, papaver roas, for example, in Turkey, contains different alkaloids compared to other papavers around the world. This study addressed the importance of conversion of wild plants in Jordan, wild lands, since it has been witnessed how fast papaver roas and even papaver polytrate can disappear from different locations in Jordan, in Amman mainly, in the last 3 years, adopting fast measures to prevent the extensions of more wild medicine plants, creating seed banks, for example, preventing the steel and concrete plants that are taking place in Wadi Shaib and also in Shafa Badran and Beli. These areas are very rich areas with wild plants. This is the end of my story. I talked to you about the Babar polytrichum, and we went through the biological evaluation and phytochemical evaluation. Hopefully, you find it nice. Thank you very much.
ما راح يجي اتصور في زميلي له راح
We can start the next speaker, Dr. Ammar Maida. Dr. Ammar is our colleague at Jordan University of Science and Technology. He received his BSc in pharmacy from the University of Jordan. Then he received his PhD from Queen's University in the UK. The title of the presentation, Morocorine, a novel cationic alpha helical peptide with cytotoxic activity against prostate cancer cell lines from scorpion venom. Dr. Ammar, the floor is yours.
Thank you, Dr. Khulud. Assalamu alaikum. Topic of my talk today is about mauroporin, which is a novel cationic alpha helical peptide with selective cytotoxic activity against prostate cancer cell line that was discovered and identified from the venom of the Moroccan scorpion A. mauritanicus. First of all, prostate cancer is considered to be one of the most common diagnosed malignancy in elderly males and the second most common cancer killer in the Western world. About 240,000 cases of prostate cancer are expected to be diagnosed this year in the United States, and around 28,000 people are expected to die as a result of this disease. Additionally, prostate cancer has become a major malignancy and health burden facing the developing countries such as Jordan. The current treatment regimens for patients with prostate cancer or localized prostate cancer include surgery, prostatectomy, plus a combination approach of radiation therapy and androgen suppression therapy. However, a big significant proportion of patients undergoing this surgery will develop recurrent disease in less than 10 years' time, about 30 to 50%. And the treatment options for metastatic prostate cancer are limited actually to mainly chemotherapy, which has severe side effects and can affect the quality of life of patients undergoing chemotherapy. So therefore, novel agents for the treatment of prostate cancer are highly needed at the moment. Let's talk about scorpions. The venom of scorpions contain a cocktail of biologically active peptide that has a purpose of defending the scorpion against predators and also helps in capturing prey for scorpions. These peptides are actually classified into disulfide-rich peptides, which usually target the membrane-bound ion channels and are responsible for the neurotoxic effects that are experienced in humans during scorpion envenomation. Additionally, there's a small group of peptides within the venom, which is called the non-disulfide-rich peptides, that display several multifunctional activities. Some of the activities displayed by these peptides include antimicrobial, bradykinin, hemolytic, analgesic, and several others. Now, the disulfide-rich peptides, the first group, contain some potent peptides with anti-cancer activity. An example is chlorotoxin, which was identified from the venom lerus. It has potent activity against glioma cell lines and is currently in phase 2 clinical trials for the treatment of brain cancer. Additionally, lately, another peptide that was isolated from an Indian scorpion called bengalin has been shown to have potent anti-leukemic or anti-proliferative activity against leukemic cells through an apoptotic mediated pathway. Now, to the best of our knowledge, there is no scorpion peptide from the non-disulfide-rich family that have been reported to display anti-cancer activities. So the aim of our project was to identify novel bioactive peptides with potent anti-cancer activities against prostate cancer cell lines through the application of molecular cloning techniques. We also wanted to identify the peptide if it was present and secreted within the venom itself of the scorpion using analytical techniques such as RP-HPLC and mass spectrometry. Once the peptide was identified, we wanted to chemically synthesize this peptide and assess its bioactivity on several cancerous cell lines and non-cancerous cell lines in order to identify if it contains or displays a selective activity against cancer. We also wanted to identify the secondary structure of the peptide and 3D dimensional structure, and also try to elucidate the mechanism of action of the peptide. So with the methods we used, basically we got the venom from a specialized company called Latoxan in France, which supplies venoms as a lyophilized powder. We got the powder, we extracted or we isolated the messenger RNA of the venom gland itself using magnetic dynabeads technology, which is, it uses beads to just separate the messenger RNA itself. We use this mRNA for the synthesis of a cDNA library representing the venom gland itself, and we have used what we call a shotgun cloning technique, which means that we have no information at all about what's in the venom itself, so we designed several primers based on homologous 5' untranslated region of several peptides within the arthropod family of animals. And we try to look or hit for peptide using what we call as race technology. This is also used by CloneTech. If we got hits, we cloned these PCR products using PGMT vector, and then we got them for sequencing. We did bioinformatic analysis, sequence similarity search, such as NCBI BLAST. We also did multiple sequence alignments using CLUSTAL-W. We also predicted the putative signal peptide. We also want to know about the post-translational modification of the protein itself. And we got helical wheel projections for mauroporin. We also wanted to assess if the peptide is present, so we did RP-HPLC fractionation for the venom itself. We fractionated the peptide. We got all the fraction, assessed their anti-proliferative activity, looking for hits, okay? And the fractions that got some kind of anti-proliferative activity, we sent them for mass analysis using Mandelbrot mass spectrometry. The synthetic replicate of the peptide was synthesized in China by a specialized company called GL Biochem, and the identity of the peptide was also additionally confirmed using RP-HPLC and mass spectrometry. We did circular dichroism analysis to determine the secondary structure of the peptide in two solvents, in aqueous solution and membrane mimicking solutions, because the solution and actually the biological membrane differs, so we used 50% trifluoroethanol. We did an in silico analysis and tried to molecularly model the peptide itself using what we call as homology modeling. And then we tested the peptide using cell culture against 3 cancerous cell line, prostate cancer cell lines, and 2 non-tumorigenic cell lines, one from humans and one from monkeys, African green monkey. We did cell proliferation assay using MTT assay. I'm not going to talk about this, most of people who who work in this area know about it. Also, we did lactate dihydrogenase LDH assay, which looks for a leakage of intercellular enzyme LDH after membrane fragmentation. So it also gives us an idea about if the peptide is actually lytic and causing membrane damage. We did hemolytic activity against erythrocytes to see if the peptide is actually toxic to blood cells. And we also tried to elucidate the mechanism of action of cell death. Was it an apoptotic or a necrotic mode of cell death using DNA fragmentation, which is one of the major biochemical hallmarks of apoptosis. And we also looked for caspase activity, which is one of the major executioner caspases involved in the process of apoptosis. So after the PCR amplification and cloning of the peptide, we managed to get a consistent single band representing 700 base pairs of the precursor of our peptide. Of course, at this stage, we didn't know anything about mauroporin or the peptide. This peptide was cloned, gel purified and cloned, and sent for sequencing. We got an open reading frame precursor of mauroporin, which consists of 73 amino acids. Just to let you know, the peptides in general, they go through post-translational modification. So the peptide is actually smaller than that. We identified the theoretical signal peptide, which is composed of 23 amino acids using certain softwares. And we also identified a C-terminal bridge, triple arginine bridge that was also cleaved during post-translational modification. And this left us with a 48 amino acid peptide. We named it mauroporin according to the name of the scorpion itself. It's a Moroccan scorpion, so a pan-Arab thing. So it's called mauroporin. Sequence homology searches of mauroporin revealed the peptide display high sequence similarity with other membrane-active peptides. So we have an idea, we got an idea about the activity. It's actually could have membrane activity, which means it could have anti-proliferative or antimicrobial activities. Helical wheel projections display that peptide is amphipathic, having hydrophilic and hydrophobic regions, which also confirms the activity prediction. We also did the fractionation, and we got a window of fraction 93 to 96 displayed anti-proliferative activity against cancer cell lines. Then we sent them for mass spectrometry. We got the mass identity of the peptides. Fraction number 95 had this identical mass to the theoretical mass we calculated for mauroporin, which is 5,000 roughly Dalton. And we knew that the peptide is actually actively secreted within the venom itself. So we confirmed that the peptide is present actually. And our prediction for post-translation and modification were actually correct. We did a cDNA analysis. The cDNA analysis in water revealed the peptide displaying a negative band at 200 nanomolars, which is consistent with peptides in unordered random coil conformation. So the peptide in water has no orientation at all. When it was shifted to 50% trifluoroethanol, the peptide suddenly changed its conformation for an alpha helical structure using CD spectroscopy. The calculation was done by a special software called K2D2, and it revealed the peptide to display 70% helicity. So it's helical, cationic, and probably membrane active. And this is also consistent with other membrane active peptides reported in literature. This shift in conformation. This is a nice picture. We did molecular modeling of mauroporin. It also displayed a consistent 3D structure. We did also confirmatory studies to analyze the structure itself. Of course, this is theoretical. We built the model based on mathematical calculations, based on other peptides with sequence homology that have been identified either by NMR or protein crystallography. The anti-proliferative activity of mauroporin, it showed that mauroporin has very high potent anti-cancer activity against prostate cancer cell lines with an IC50 average of 7 micromolars. So it was showing anti-proliferative activity against cancer cell lines with very low or potent activity. We did also the anti-proliferative activity against normal cells, and the IC50 average was around 70 or 60 micromolar. So there is a big gap, or let's see, selectivity index. It's killing this cancer at 7, while the normal cells are being killed at 70 micromolar. We confirmed this by LDH release, membrane disruption. Also, the peptide is showing selective activity against cancer cell lines with no damage to normal cells at the same concentration. We also tested the hemolytic activity We did a time course for mavacoric during several time points to see if the peptide is actually displaying fast killing kinetics. So the kinetics showed that mavacoric will kill 50% of the cells, cancer cells, within 3 hours, reaching maximum killing rate at 6 hours. We did hemolysis. At the concentrations needed to kill cancer cells, the peptide caused around 1.7% hemolysis, which is insignificant. At 80 micromolars, which is 10 times higher than the IC50, it caused only around 4.8 micromolar percent hemolysis. We looked for DNA fragmentation. The peptide was not causing DNA fragmentation, so it's not causing apoptosis. No increase in caspase 3 activity. I know this is negative results, but the trend in literature now is to present negative results because they are also significant. So in conclusion, cancer is still a major problem facing the global population. Most cancer treatments focus on chemotherapy, which has very bad side effects, and also cancer cells can develop resistance against chemotherapy. The cationic alpha helical peptides, such as mavacoric, do not have the same toxicities as chemotherapy, and also they do not have an intracellular mode of cell mechanism, because our data displays that peptide is actually causing membrane damage. It's a lipopeptide with selective activity. So mavacoric could be a potential candidate for drug development as an anti-cancer agent. Of course, several studies are needed to assess the safety of mavacoric in biological models, but our initial data confirm the activity of mavacoric against prostate cancer cell lines. Thank you for listening.
Our next speaker is Professor
I need just to brief acknowledgement. This project was my first project at JUST, and Dr. Khulud Al-Khamis had a major contribution in fighting for the budget in the Dean's Council. Dr. Bashar Ta'ani also supported me logistically, so I need to acknowledge this. Thank you so much.
سيارة مرسيدس بيضاء لجامعة الهاشمية، شباكها مفتوح وفيها لابتوب.
الكل سمع الملاحظة، السيارة البيضاء عليها ستيكرز الجامعة الهاشمية، فيها شباك مفتوح وفيها لابتوب، فاللي إلى السيارة يروح. تفضل دكتور.
Unfortunately, they did not provide me with a CV, so I will ask Doctor to introduce himself. Our next presentation is the future of genome medicine in patient care for Professor Hakonarsson from the University of Pennsylvania. Doctor, could you please say a couple of words about yourself? Sorry.
Well, thank you for that introduction. So I'm from Iceland, and I graduated from the Medical University of Iceland, where I did both my MD and PhD. I trained in the US, residency at the University of Connecticut, and then fellowship at Children's Hospital of Philadelphia. And I was an associate professor at University of Pennsylvania for a couple of years, then moved back to Iceland, and I was director of this big genomics project in Iceland of genotyping the entire Icelandic nation. And then I moved back to the States in 2006, and I built a genomics program there. And I'm going to tell you a little bit about one of the programs that we are taking forward into clinical development based on genomic discoveries. So this is now up. Let me see. So I presented the first two slides in my earlier talk, but I think most of you are a different audience. Just to give you a very brief background, I built this program in 2006, and we have now grown up to a staff of 75 We are working on over 60 different diseases. We have genotyped over 100,000, recruited over 100,000 individuals who are children. 70,000 of them are linked to electronic medical healthcare records, and we have about 220 or so thousand samples in our biobank currently, as shown on this slide. And you can see that we have a lot of healthy controls, and we have big sample sizes across all disease areas, and we also have a lot of rare diseases that we have been working on. Now, one of the sort of flagship project that we have done is a neurodevelopmental phenotyping of 10,000 patients that we have recruited and we have genotyped who came back for comprehensive neurocognitive phenotyping. And this was a very, very big study funded by the NIH with about $20 million. And we did battery of tests. We did MRI studies on about 2,000 individuals, and we have genotyped everybody, and all of these individuals have given us opportunity to sort of recontact them for other studies. And this is just a schematic overview. There's a clinical assessment, and then there is this cognitive battery of tests, which takes a couple of hours to do, and this was done on all 10,000 individuals, and then 2,000 have the imaging, and all of these patients have been previously recruited, genotyped, and we sort of screened out of the cohort we had individuals who were almost like a population-based cohort, because we recruit patients and healthy controls from about 20 different clinics all over the Philadelphia area. And when you do this type of analysis, you can actually do a phenotype classification of the individuals. And anxiety disorders, behavioral disorders, psychiatric disorders, and psychosis, addiction, eating disorders, a whole host of things that you can sort of analyze from the spectrum of the assessment when you take the top 5% of individuals in each category. And we have now taken this forward and sequenced these individuals for molecular phenotyping. And one of the projects that came out of this is in ADHD. And ADHD, as you know, is the most common of the neuropsychiatric diseases. And it's actually very undertreated because there is a stigma associated with the stimulant medications and some of the norepinephrine uptake and receptor inhibitors and adrenergic drugs. They have significant side effects, so people don't want to take them. And but children who have this condition, they are kids who drop out of school, they are kids who end up in conflict with the law, so it's a very, very costly disease which is very under-treated. And I believe it's no different in other countries than it is in the States and Iceland, as I perceive them to be very similar there. So what prompted this was the discovery of copy number variants, and I explained in my earlier talk what copy number variants are. This is basically deletion or duplication of a region of the genome, and in this instance we found that patients with ADHD, they were heavily enriched, about 10-fold over and above controls, with copy number deletions and duplications across genes that belong to the metabotrophic glutamate receptor signaling pathway. And as you know, glutamate, which is a neurotransmitter in the brain, is really important for memory, attention, behavior, and various cognitive functions. So this sort of turned out to be important, and this was totally hypothesis-independent approach. So therefore, we did not pick these genes, they just came up because they were the most significant in the study. And then we looked at them more carefully, and we found that there were actually over 60 genes that were significantly enriched in ADHD. And GRM5, GRM7, 8, and 1 were actually the most significant ones, and they are sort of the big diamonds here, and the big gray ones here are GRM1, GRM4, 3, etc. And so it was very interesting that all of these GRM genes, they are actually activated by glutamate. So glutamate acts through all of them. And then when we sort of looked more carefully, and this is just sort of, as I said, classifying the lead GRM1, 5, 8, 7, sort of as the lead genes. There were other genes in the same signaling pathway that showed copy number deletions. And when we did a more high-density array, we found that there were somewhere between 15 and 20% of kids with ADHD who have these deletions. Now, this was sort of interesting because there was a drug that a Japanese company had developed for a different indication, but they had done all the phases of the studies, phase 1, all preclinical studies, phase 1, phase 2, phase 3, and they did not reach the endpoint of success, so they put the drug on the shelf. But the drug is a derivative of glutamate, and the drug actually is a glutamate agonist. And so the notion here is, well, maybe patients who are missing one copy of a gene, the drug can come in and work and make the other copy work twice as hard and normalize this. So that was sort of the hypothesis. And the Japanese company had actually seen cognitive improvement in animal studies by giving this drug. But when we go back and look at these studies, and we've repeated all of them, we think it's sort of more perhaps attention improvement rather than cognition. But that can be argued because, but animal studies obviously are never quite human studies. But it's very easy for us to identify the patients who have the deletions because we have genotyped all of them and we know who they are. And the drug, as I said, works on all of the same receptors as we found to be deleted and altered in the patients. And interestingly, there are independent studies that have looked at some of these GRM receptors. GRM5 was the most significant in our study, and if you knock GRM5 out in animals, the animals become hyperactive and they become inattentive. And if you give an inhibitor drug, you see the same dose-dependent effects on the animals, suggesting that this receptor is important for activity and attention. And same holds true for GRM7 and GRM8. You get the exact same information, and these are done by totally independent laboratory, have nothing to do with us. And when we did similar studies to what the Japanese company had done in rats to show that the drug impacts on and reduces hyperactivity, we got the exact same results in a dose-dependent way. So this was highly, highly encouraging and sort of suggests that we might be able to take this drug and now treat children who have one copy and are missing another copy and make the drug correct the physiological condition. It's almost like giving a thyroid hormone when you are low in thyroid. So we're not really giving a drug that has a lot of side effects because it's almost like a same as glutamate. And so it's a very significant sort of risk reduction because the compound had been treated all the way through phase 3. And so if you think about it sort of from the standpoint of stimulant drugs and the norepinephrine receptor uptake inhibitors and adrenergic drugs that I mentioned, which are sort of not highly selective and not highly effective, work for the stimulants maybe for about 55% of patients, we would envision that this drug would be both very selective and effective in the patients who have the mutations. And that would be our goal. And if you think about it sort of in the context of previous examples, I'm sure most of you are familiar with the drug Herceptin, which is a drug to treat breast cancer. And Herceptin is a very, very effective drug if the breast cancer expresses a protein which is called HER2. If HER2 is not expressed, there's no effect from it. So our thinking here is that our drug, which we refer to as fasuracetam, because it's an acetam drug, if there is a mutation, so you have only half of the material in the brain to keep you alert, to keep your memory normal, and keep your activity level normal, if you are missing that copy, you could give a drug that reverses that. But if you don't, then you have nothing but these non-selective therapies which have many, many more side effects, like the chemotherapy and radiation if Herceptin is not working because there's no HER2 protein. So this is sort of, I mentioned this example in my other talk before, that we have previously basically identified kids with a very rare form of neuroblastoma, familial form of neuroblastoma, and we found a drug that basically melts the tumor away in these kids. And now, that's obviously is a rare cancer, but now we have a much more common disease, which is ADHD, which is present in about 8% of the population sort of across the world, where we can take a drug which has previously failed for the studies of dementia, but now treat ADHD, which are present in individuals who have these copy number deletions. And I put here autism and schizophrenia because patients with autism and schizophrenia also have deletions in this same signaling system. So we don't have to look for patients because we know exactly who they are in our database. We have 200,000 samples there, they're all genotyped, and so we can now take this, and we are now working on filing an IND. We got all the data from the Japanese company, so we are now taking this into clinical trial, similar to the neuroblastoma trial, which was very, very successful, and we envision that we can get this, if this works, out in about 46 months or 4 years, whereas conventional drug development, as you know, takes about 12 to 15 years. So it's obviously a huge, huge advance, and it treats the specific cause of ADHD in these kids. And this is all driven by our genomic discoveries. And at the same time, we are doing studies on iPS cells. So we take the blood cells from these individuals who have copy number deletions versus not, and we look at neurons that we basically derive from the iPS cells. So iPS are induced pluripotent stem cells that we basically transform or differentiate into neurons, and the neurons have these receptors, so we can study these receptors in the neurons. So this is a fascinating program coming out of the genetics, how you can use genetic markers to select a specific population which is going to respond specifically to a certain medication. And this is really sort of the pharmacogenomics model. We have a neurological disorder, and as I mentioned, we will go to autism and schizophrenia next if this works, but we have identified copy number variations. These are single nucleotide polymorphisms. We know exactly which one we need to test for. They basically identify a network of genes where the metabotrophic glutamate receptor pathway is involved, and we have a drug which is an agonist that can fix this, and we can sort of fast track this to a clinical trial, and hopefully a successful one in a short period of time. And with that, I was going to stop, but there's a lot of people in my group who work with me, and I want to thank you for the attention. Thank you.
Our next speaker is Professor Jolanta Gutkowska. Professor Jolanta is from Yale University, Experimental Medicine. She is the Director of the Laboratory of Cardiovascular Research. Professor Jolanta, the floor is yours.
Thank you very much. I'm really extremely pleased to be here. And you know, after my first presentation, I got several extremely nice comments. Unfortunately, do you hear? Yes? Is it okay? Yes. Oh, I paid already. I paid already. Yes, the person returned. I'm sorry. So I will continue. presentation, my conference was cut to about half. Not because I was really talking and developing very much the subject, but my previous speakers took too long time. So since the comments which I got were very nice and the people were interested, I would like to repeat this conference, giving you much more details on the part of my speech, which was not delivered. I hope that you don't mind, but especially I think it is very important. I see a lot of students that this work which I am presenting, it is very systematic studies, and the message is that really, if you have some interesting subject in your hands, you shouldn't change, you should follow. You know, from a very simple experiment, we reached the cell therapy. It is what I would like shortly to develop today. So, all of you were at the morning presentation? Yes? Well, so I will be very brief. Excuse me? Yes, it is the painting, you know. I'm working in the first hospital which was created in North America by the French nurse Jeanne Mans, and it is the old part of the building. Now we are moving to super modern 2020 building, which is very well prepared for the very modern research. So how should I change? Okay, I would like to, as I told in the morning, I would like to tell you a very, very new development in the cardiovascular system. Certainly you all know that heart is not really the pump. It is not the only role of the pump which delivers the oxygenated blood in the lungs. But also it is an extremely important endocrine organ. For the first time, it has been shown in 1981 by Professor De Bolt, who is Canadian. He showed that heart produces an important hormone, which is a very potent diuretic and natriuretic. So today I learned something very interesting. This Aristotle wrote about 400 BC that heart is a center of thought. The brain contributes to the vital fluid circulation. We know that the second thesis has been already proven. We are working on the first, but what I learned today, that Prophet Muhammad also wrote about or talked about that the heart is the center of thought. You know, oxytocin has been known more than 100 years, and it was traditionally considered as a hormone which is involved in the regulation of reproductive function, ovulation, lactation. But the last 2 decades revisited this idea, and oxytocin is involved in several regulations of neuromediated function. It is involved in the maintenance of relationship between the people. Apparently, oxytocin may involve the relation and the trust among the people. It is involved in the food and the drink intake, effect of cell proliferation, and recently, during last years, we have shown that it is also play a very important role in the cardiovascular regulation. So, as I mentioned in the morning, that in the 60s, several groups of people were working on the stimulation of different area of brain, which produces diuresis and natriuresis, but there was no any explanation how the brain may rule the kidneys until what I just mentioned, the discovery of natriuretic peptides. It is the family of peptides very short peptide, 17 or 20 amino acid, which injected into the circulation induces a very potent diuresis and natriuresis. So the idea was that probably during this mechanical or chemical or pharmacological stimulation of the area of brain, there is a release of ANP and diuresis which issue. But it was not conceivable because the quantity of ANP are very small in the brain. And when we repeated the experiments which were done originally in the 60s, we stimulated the third ventricle, which is a very key area, with carbachol. We observed the release of ANP, but this release was huge. It was from about 50 or 25 to 2,000 picogram per milliliter. It was not conceivable that the brain may produce and release such a huge quantity of ANP. When the median evidence, the area of ANP were electrically, there was disappearance of ANP. But this ANP which is released could not be coming from the brain. Therefore, we looked for alternative. What is the mechanism of this ANP release? And we discovered the neuronal circuit which exists during the volume expansion. Volume expansion was one of the most potent stimuli of ANP release. So we used animals which had volume expanded, and we blocked the different pathways like baroreceptors, atrial receptors, the serotonergic system, cholinergic system, and we found finally that in hypophysectomized animals, where there are two hormones, oxytocin and vasopressin, if we did volume expansion in these animals, there was no release of ANP. Therefore, when we later on performed only lobectomy, it means pars nervosa, neural part, there was no release of ANP. Therefore, the conclusion from this was that probably one of the hormones which is present in pars nervosa, oxytocin, all vasopressin, are released. Act probably on the heart, which was the source, the most important source of ANP. ANP is released and diuresis. So, vasopressin could not be our candidate because vasopressin at this moment has been already shown to release ANP, but only via hemodynamic changes. So another candidate was oxytocin, and we injected oxytocin in the rats and observed the dose relation release of ANP and the same natriuresis and diuresis. So if we look at this diagram, so we see that oxytocin could be released from the neural lobe to the circulation and acts on the heart, but if it is true, the heart should contain the oxytocin receptors. And indeed, by the different methodology, by cytohydratization, by autoradiography, we found that in those ventricles and in those atria, there are receptors for oxytocin. So to eliminate the neural system, we performed perfusion of heart and used oxytocin as a stimuli for ANP release. And this is very important slide. We published this in 1997 in PNAS. As you see, oxytocin is really a very potent stimulant of ANP, and it is low stimulation. But oxytocin antagonist not only inhibits the oxytocin-induced AMP release, but also control on the spontaneous release of AMP, suggesting that oxytocin is also synthesized in the heart. And indeed, in around